Hi guys. So I just wanted to round off our talk and lecture about the carbohydrates just by talking about these modified carbohydrates. And these are where the alcohol functional groups maybe get changed to something else. And if we start, for instance, with glucose here on the left, and we have all of the different alcohol groups, some of these may get swapped out for other functional groups. For instance, this one here might get swapped out for an amine. And this would produce, since it's started off glucose, of the root glucose, it'll produce glucosamine. So it's glucosamine, glucosamine. You may have heard of glucosamine, for instance, as a supplement that you can buy in stores that is often referred to as being for joints and pains and things like that. And that's going to come up relevant later. Or you can have the acetyl. So here, acetyl or acetyl glucosamine. You can have as well the glucuronate, which is down here. This is the glucose, but it has this carboxylic acid down here, so it's glucuronate. Or even if you have galactose sugar, where it's got a different orientation of those OHs, then if you've got the amine of the galactose sugar, then it'd be galactosamine, mannose-amine, and things like that. Or when we're talking about deoxy sugars, like as in deoxyribose nucleic acid, where you take your OHs and maybe you swap it to remove it or swap it out for a CH3. So these all kind of the modified, they're slightly different modified sugars. Sometimes I like to refer to them as being heterosugars because they're different sugars the functional groups have been changed. Now, this comes up in is important in biology in a few different ways. For instance, the glycosaminoglycans. If we break that down, glycosaminoglycans. Well, glycans is an old-fashioned biologist way of saying polypeptide. So it's a chain of sugars. And glycosamino, we know that those are those sugars, but we're swapping in the amines. So these are going to be chains of sugars that have got these glycosamines in them. And these come up a lot in biology, for instance, for often they come up as these kind of dipeptide repeating units. So if it's kind of sugar 1, sugar 2, and it'll be a pattern of 1, 2, 1, 2, 1, 2, 1, 2. That's a repeating disaccharide pattern. And these come up a lot when we're talking about hydrating molecules. Hydrating molecules, particularly for the extracellular matrix. So you have in cell biology, you have your cells, but surrounding your cells is often a kind of gooey kind of matrix, hydrating gooey matrix that kind of sits around the cells, helps making up the connective tissue, and helps provide a lubrication of your joints. And you kind of go over more detail of that in anatomy and physiology. But really, it's like I say, it's just a something that's extracellular, so it's outside the cells. Matrix is like a mixture of things, proteins and sugars, and these glycosaminoglycans. They all come together to try and make a gooey, hydrating matrix to kind of help hold everything together. And you get these glycoaminoglycans. These are kind of handy because these amine groups or carboxylic acid groups, you're introducing additional hydrogen bond acceptors, hydrogen bond donors, or these negatively charged groups. They're going to attract water. And so they end up being very hydrating. They bring in water to help keep everything all, like I say, lubricated, or to help insulate it, to help hydrate it as well. And some of the most common ones that might come up are going to be things like hyaluronate, as in hyaluronic acid, which you may recognize from being in moisturizers. And this is a glycuronic acid and acetyl glucosamine. So here is the carboxylic acid, is the acetylamine. And these are GLC, so that's meaning that it's glucose. Glucose with the acetyl, the amino acetyl, or with the acid, the carboxylic acid. And it's that 1, 2 repeating pattern, making hyaluronic acid very hydrating, because that carboxylic acid and that acetyl amine, that is going to attract lots of water and end up being very hydrating. So it's a hydrating molecule in moisturizers. It's also a hydrating molecule in your body as well, and as well in this one as well, where you have the glucuronic acid, but also you have and you have your acetylamine. Also, you have this sulfuric acid as well, and this is also going to be very negatively charged. It's going to attract a lot of water, and it's going to be very, very hydrating. So these are very hydrating molecules. They're repeating. The glycosaminoglycans repeating 1-2 pattern repeating disaccharides. Moisturizers, lubricators, hydrators in the extracellular matrix. And so I kind of talked about a lot of this stuff. When you see these sugars attached to things, and you know what I've mentioned with the glycosaminoglycans, that comes up a little bit more later. But I've talked already when we talked about proteins that you can glycosylate them. You can add sugars to proteins, or lipids. We can add sugars to lipids. And you remember with the blood typing, and you had the sphingolipids, and you had the different combinations of sugars, and that makes the different blood types. Or even identifying a human versus another human, a tissue versus another tissue, or even between species, between a pig heart and a human heart, it's going to have different glycolipids, glycoproteins, which are going to be that identifier. Remember that sugar code. And where you have the proteins, you can have them glycosylated, so you can have a protein with a little bit of sugar, or these proteoglycans, which really you can kind of think of as being a lot more of based around the sugar, and the protein really is just a scaffold to hold the sugar, actually more so. And this is kind of summarized across in this table here, but the proteoglycans, like I say, they're kind of like a protein scaffold with sugar attached to it. So the sugar is really the thing that's important, and the protein's just there to hold it still and to hold it into place. I've got a picture to show you guys later. But the glycoproteins are really where the protein is the main thing, and the sugar is something that's just added on to maybe affect it slightly, maybe to act as that identifier. So it's whether or not the sugar is the main thing, or whether or not the protein is the main thing. Or with the glycolipids, where it's a lipid with a sugar attached to it for identification. And for instance, a glycoconjugate, really, these all belong to the family of glycoconjugates, where it's glycoproteins, proteoglycans, or glycosphingolipids, or glycolipids, glycosphingolipids. The difference between glycoproteins and proteoglycans is something that I'm not really going to test very much in Biochemistry 1, but really to be aware that proteins can be glycosylated, or that lipids can be glycosylated, and these are glycoconjugates. Now, for the example of the glycolipids, these came up, for instance, with the blood grouping, like I talked about, with the sphingolipids that have that combination of sugars that makes you blood type A or B, or if you have both of them, you're AB, or if you actually are missing the terminal sugar, you're going to be blood type O. I mentioned that in the lipids lecture. Another example are these lipopolysaccharides, also known as LPS. And these are lipids, membrane lipids, you can see here. These are membrane lipids, and these make up the bacterial membrane. But these are lipids that make up the bacterial lipid membrane. But in gram-negative bacteria, they're going to be glycosylated, having a chain of sugars that can be very long. Let's go into identify the bacteria as being a bacteria. Like I said, it's that sugar code, it's that identifier, it's that combination of sugars that lets us identify things. And actually, this particular one here is common in gram-negative bacteria. It's called LPS, lipopolysaccharide. Lipo is a lipid, polysaccharide is a chain of sugars. And your immune system can recognize LPS, and it'll say, oh, there's a bacteria here, and it will stimulate an immune response. LPS will stimulate an immune response because that chain of sugars is like a signature for a bacteria being present, particularly gram-negative bacteria. On the other hand, the glycoproteins, like I said, that is where a protein can have a protein, amino acid of a protein can have a sugar attached to it. And that sugar may be fairly long or short, but usually with the glycoproteins, it's really where the protein is the important part, and the sugar is just something that's added on just to modify the protein's function. Really, the sugar is modifying the protein with the glycoproteins. They can be O-linked or N-linked, which is really where they link, example with serine and threonine, which are amino acids which have got an OH group in their R group, and the oxygen of the OH group will attack that anomeric carbon and cause an O-glycosidic bond, so it's called O-linked. Or if it's the amino acids that have got nitrogen in their R group, where it can come in and it can attack, the nitrogen can attack the anomeric carbon, just like the nitrogen is attacking the anomeric carbon in the bases that make up the nucleosides, where it attacks and links the base and the sugar together for a nucleoside. Remember, the nucleotide is when you then add on a phosphate to the sugar as well. But this is really where you've got a protein, an amino acid, and you're just modifying that amino acid, adding a sugar onto it to modify its function for whatever reason. And this is often for things, well, with the lipids and with the proteins, this is often for recognition. This is often because that combination of sugars is going to attach onto the lipid or attach onto the protein. And different receptors are going to recognize that combination of sugars, and it's going to signal that something is happening. And it's going to signal that something is present, and it's an identifier that is really, really important in biology. Or for instance, in the immune system, you can have your leukocytes are flying around, whizzing around in your blood, and they don't know when to attach. But what will happen is that if your blood vessels need a leukocyte to attach, so it can have elicit an immune response to whatever is happening. Maybe it's an infection or something like that. Maybe you've got an infection that's causing inflammation, and your vascular cells will release glycoproteins, the proteins that have got that sugar signal, and that sugar signal is going to say, come here. And your leukocytes are flying around, they attach to one of these glycosylated proteins that's been released as a response to inflammation, that's going to say, hey, stop here, and then slowly come in, attach, and attack the site of inflammation because there's an infection here or something like that. The other one, like I mentioned, was the proteoglycans. That is where the sugar is the thing doing all of the work, and the protein is really just a scaffold. So you can see here that the protein is just a scaffold, and the sugars are attached to that. And these are where you're going to get those glycosaminoglycans, those very hydrating molecules. Those very hydrating glycosaminoglycans are going to attach to proteins as scaffolds to pull water into the area. So those glycosaminoglycans attract a lot of water, but they're just going to float around, and so they'll be anchored into place by proteins. So it's the sugar that's doing all the work, the sugar that's important, the sugar that we're focusing on, and the glycosaminoglycans, and this shouldn't have the SE in it. Glycosaminoglycans are the ones that are attaching to the protein, and the protein's just holding it into place. And these can actually form very, very complicated structures. They can be very big, and they can hold a lot of water. For instance, hyaluronic acid can be huge molecular mass, and can hold an amount of water 1,000 times its weight, thus providing a large amount of lubrication to the area, and in particular is very common in cartilage and joints. But and this is what it might look like, for instance, is that you've got this, let me change color with the pen so it shows up better. You've got this long protein scaffold, and then you've got other proteins even coming off that scaffold to make it more branched. And then on each of the branches, you've got these glycosaminoglycans making a really big surface area so that it can attract lots of water. And it's going to look something like that, actually, if you were to look at it down a microscope. And think how large that must be if it can be seen by, I think this is an electron microscope, but still, it's a fairly large structure. This is a huge number of sugars chained together, attached to a protein that may be anchored onto a cell membrane. and it's going to attract a lot of water. And that really summarizes what I wanted to tell you guys about these these hetero sugars. The idea that you have normal sugars, you can change the functional groups, and for instance, to an amine, to a carboxylic acid, to a acetylated amine, a secondary amine, all sorts of different things. You can take away the OH, replace it with a CH3 methyl group, all kinds of things. There's lots of different reasons why you would do that. One of the most common ones is for attracting these groups, having on these amines and sulfates and carboxylic acids that attract water, and then you can add them to proteins, and they can be in the extracellular matrix to act as a hydrator for the extracellular matrix. Just remember the glycoconjugates for the glycoproteins, the proteoglycans, such as this is a proteoglycan, or the glycoproteins, or the glycolipids. And that's really just covers the last little bit I wanted to cover of carbohydrates. Thank you for your time and attention.